Good morning. So today's topic will be based on embryonic development. So embryonic development, first of all, what is embryo? Embryo is the developing stage. It is not the complete fetus. It is the developing stage when an egg is fertilized with a sperm. The fertilized egg, what is the development taking place inside the uterus wall? That is the full study of embryonic development, and we can say this study as embryology. The embryonic development, also known as embryology, the study of embryo. Now the first thing happening is an egg or ova. It contains n number of cells. It is a ova or egg cell. When sperm is coming, which is also carrying n number of cell, n number of chromosome. When it is fertilized, when it is penetrating and when it is fertilized, now it will become 2n number of cell, and this we call fertilized egg carrying 2n number of chromosome. This fertilized egg, we also say the fertilized egg as zygote. Now after the fertilization, this cell process through a number of developmental process. It undergoes a number of developmental changes. First of all, cleavage takes place. Cleavage takes place. What is a cleavage? Is this cell is surrounded by a layer that is ZP or zona pellucida. Zona pellucida is protecting the egg from the external resistant. It acts as a barrier, and it also contains a lots of floating cell around the egg. This we call as corona radiata, because it is radiating. That's why corona radiata. Now the cleavage. The first cleavage is happening after 30 hours. The first cleavage is happening after 30 hours of fertilization. Now this fertilization is taking place on the ampulla of oviduct. This is the location of the fertilization of egg and sperm. This is ampulla of the oviduct. In this region, this is the ovary. In this region, this is the ampulla of oviduct. Now after 30 hours of fertilization, first cleavage happens. In the first cleavage, what happens? A 2-cell stage form. Like this is the whole thing, and there will be 1, 2 and corona radiata as well as the zona pellucida will be remain there. Zona pellucida, throughout the cleavage, zona pellucida will always protect the fertilized cell. Now this becomes 2-cell stage. This is a 2-cell stage. This is after 30 hours of fertilization. Now after 30 hours, the 60 hours of fertilization. After 60 hours of fertilization, second cleavage occurs. And in the second cleavage, the first 2 cell will again divide, and it become 1, 2, 3 stage. This takes place at the 60 hours after fertilization, that is 3-cell stage. Now after this, after the 3-cell stage, there will be a third cleavage. The third cleavage is longitudinal in nature. That is 1, 2, 3 and 4. Now it will become a 4-cell stage. This is 4-cell stage. Very important point. Now it become a 4th-cell stage, and now the cytoplasmic content will remain same. The cytoplasmic content of the whole zygote, what we are telling, will be remain same, but the nuclear content will not be remain same, because when ova was there and sperm fertilized it, it become 2n, right? But now it has become 4 cell. So 2 into 4, it become 8n. So nuclear content will not be same, and in the 4-cell stage it become 8n. But the cytoplasmic content of the whole body, this thing, the cytoplasmic content will remain same. It will not change. So this is the 4-cell stage. This is the 4-cell stage. Now what happened? This process is complete. Now morulation takes place, or morula. This is number 2 is morula. Number 1 was cleavage. Till 4th cell we have learned about cleavage. Now 4th cell is there. Now in morulation is the formation of the structure, that is we called it as morula. The morula is the term which is came from mulberry, is a fruit. From the term mulberry, morula is discovered. Now this is the formation of morula. Now morula is nothing but it is now the rapid cell division taking place in morula. In morula there is a rapid cell division taking place. Now this thing is dividing and dividing, and now in morula what we can see, the cells are surrounding around. (32 to 64 cell it has become, and the outer cell we call it as tropoblast. In future it will become tropoblast. Right now it is not tropoblast. In future it will become tropoblast. We will see that in blastocyst or blastulation. But in morulation, this is the formation, and this becomes inner cell mass in the future. This we have done. So cell is arranged in a compact way. This we called, the cell is arranged throughout. The cell is arranged through the fertilized zygote. We can say that this is, we call this as tight junction, and inside this future it will become embryo, and this becomes the extra embryonic membrane, or we can see placenta, the tropoblast or the outer cell. This becomes placenta, and the inner, the most important, in future it will become the embryo and then fetus. So this contain a tight junction, and inside the cell which is arranged, it is also connected through gap junction. the gap junction is necessary because gap junction is necessary for the exchange of gases, and also it provides nutrition. So the outer cell is providing nutrition to the future inner cell mass, because this will become the embryo or fetus. So it is providing nutrition, and the gap junction is responsible for the exchange of gases. So this is the morula stage is completed. Now on number 3rd, it is blastula. Now what is blastula? Blastula means the formation of blastocyst or blastula. That is blastulation. Sorry, it is blastulation. This is the process, and this is formation of blastula and nothing else. So blastula is nothing but, if this is a cell, this is arranging properly the outer cell. This outer cell, now we call this as trophoblast, and inside there are cells. Inside there are cells that we call inner cell mass. This is blastula, and the cavity is we call blastocele. Is a cavity. It is a cavity which is providing nutrition to the ICM or the inner cell mass. This inner cell mass future becomes the embryo. Now this trophoblast is completed and ICM is completed. So this is again 32 to 64 cell stage. This blastula undergoes development, and the whole structure forms. Now inside the blastula, we can see here on this part there are cells of rubber, or we can also say as glue cells. This glue cell is responsible. Why blastulation is important? Because after blastulation, implantation will take place. Implantation takes place in the endometrium wall. Implantation takes place in the endometrium wall. So fertilization takes place here and implantation will take place here. This blastulation or blastocyst will implant on the uterine wall. Now it has a like this cells of rubber or glue cell. This glue cell is responsible for the attachment to the endometrium, and this glue cell provides an enzyme or gives out enzyme that we call plasmolysine. Plasmolysine is an enzyme which will penetrate or it will rupture the endometrium, and also this plasmolysine will rupture the zona pellucida, because in morulation also ZP was there, cleavage ZP was there, but after blastulation the formation ZP is not there. ZP will break. So zona pellucida layer around the cell layer breaks in the fertilized egg. It breaks. So endometrium, it will attack or it will implant inside the endometrium. Endometrium will rupture or break and it will place a permanent place this region, on this region. So this we called as blastulation or implantation we call. So this is the thing. Now implantation will take place after blastulation, implantation takes implantation means it will implant itself. The developing embryo will implant inside the endometrium wall. Okay, it is implanting itself. It will further become like this. This we will call it as embryonic villi, which is a finger-like projection, and it is actually the embryonic villi of the embryo or developing this blastocele. It is a junction, it is a finger-like projection which is matching or it is connecting with the uterine wall. So it is a finger-like projection. So now it is fixed that inside the uterus it will develop, it will grow, and in future the embryo, this developing embryo will become fetus, and then a perfect miracle will happen and a baby will be born. So this is the process of implantation, and this trophoblast is very important, that this trophoblast secretes a hormone that is AGC, that is human embryonic gonadotropin hormone, which plays an important role in pregnancy test, or what we call as graviton test, because this trophoblast release this hormone which confirms that the particular woman is either pregnant or not, because the release of HGC hormone, that is human cryogenic gonadotropin hormone, is released. So I hope you understood the whole process of the embryonic development, and the process starts from the fertilized egg to cleavage, morulation, blastulation and implantation, and over there it develops. Thank you.